Hey, good evening, students. Welcome back. We were revising. Yes, we were revising our chapter biomolecules, and we have revised with our enzymes, proteins, amino acids. So here today I'm going to discuss few simple tricks for the carbohydrates. I'm going to teach you about the glycosidic bonds. Many of you, they have fear in your mind that how to make this glycosidic bond, as well as I'm going to teach you structure of glucose, fructose, as well as how to draw ribose. Very basic things in this lecture. Very short video I'm making this today for just for the basic things, right? When we discuss biomolecules, now many students they feel so they do not take interest in reading this chapter because there are lots of structures. As you know now, so we made very easy how to draw peptide bond. Now you can easily draw. We made very easy how to draw primary, secondary, tertiary and quaternary structure of protein. Same way we are going to learn carbohydrates today. As you know that carbohydrates, simple sugars. I'm not talking about the cellulose. I'm not talking about glycogen. I'm just talking about the simple sugar. They are our microbiomolecules, plus they are our organic compounds. कौन से होते हैं organic compounds? Those are having carbon, right? So first thing, these are the basic thing which you should know, that simple sugars, they are micro ones, they are microbiomolecules, as well as they are organic compounds with carbon, hydrogen and oxygen. Now, from where do you get carbohydrates? From where do you get carbohydrates? What you eat? You eat potato, you eat banana, apple, corn, rice. These all are rich source of our carbohydrates, and generally, or I will say major source of carbohydrates is plants. Cane sugar, glucose, starch, they all are rich source of carbohydrates. General formula for carbohydrates is C6CX water. Why? Right now, that is the reason they are also known as hydrates of carbon. You just put 6 carbon. Suppose I just put 6 carbon, C6H12O6. I will be able to form my glucose. So basically, when we ever ask to children that what are carbohydrates, they think they just give me answer, ma'am, they are very rich source of energy. But what are carbohydrates chemically? They are organic compounds. They are organic compounds. What do they have? Carbon, they have hydrogen, they have oxygen. And if I'm talking about, if I talk about simple sugars, then they are very sweet in taste. So basically they are also known as saccharides, because those compounds which contain sugar, they are known as saccharides. Why do they name as saccharides? This word we are carrying from the Greek language, that is saccharon, and saccharon means that is sugar. Basically, that is why your simple sugars, your glucose, fructose, they all are our sweet sugars. So most of the carbohydrates, they are sweet in taste, therefore we have given the second name, the saccharides or the sugars. The most common which are mentioned in our NCERT is glucose and fructose. Okay, the one thing, major thing, when you draw the structure, there is a group here. Can you see this group? CHO. This is aldehyde group. I will say free aldehyde group. Where? In your glucose. Look here. C, that is C. Definitely it is C. This is HO. This is our ketone group. This is ketone group. So this glucose, if you look at this glucose, what kind of structure it has, what kind of group it has. Basically, first difference, that is the CHO. It has aldehyde group, and in fructose it has keto group, right? That is the major difference. Here is aldehyde and keto group. Then there is the orientation. Here HOH, OH, that is at the third carbon, that is the reversible. So alternatively OH and H group here. In third position, look at the third carbon. There is OH, H, but in fructose it is HOH. So now come at the ring structure. Glucose has 6-membered ring and fructose has 5-membered ring, right? So if I just give them numbering, so let's give them numbering 1, 2, 3, 4, 5, 6. Here we are having. So when we number our fructose ring, this is our first, second carbon, third carbon, fourth, fifth carbon, and here is our sixth carbon, right? And that is really going to help you to form your glycosidic bond. So this is the structure of glucose, and here we have fructose, 6-membered ring, and here is 5-membered ring, and here is the open chain structure, right, which is having our aldehyde group, and here it is having our ketone group. So these are the things you need to understand when you draw structure of glucose and fructose. Now comes to the another thing, that monosaccharides, they usually either they have free aldehyde group or they are having free keto group. Now I am going to introduce you to two main things, that is hydrolysis as well as dehydration synthesis. See, can you break down monosaccharides into further simple units? No, because they are the most simpler one. Simple sugars, they are the most simple units. You cannot break them. So you cannot break down fructose further, glucose, ribose, glyceraldehyde. These are monosaccharides. But yes, oligosaccharides, generally we write 2 to 7 and we exceed it to the 10 also. So carbohydrates that is made up of, listen carefully, that carbohydrates which are made up of 2 to 10 monosaccharides, means suppose 2 glucose, or you can say fructose plus glucose, right, or many units of glucose. So these are known as our oligosaccharides. They can be 2 in number, they can be with the 3 monosaccharides, they can be with the 4 monosaccharides up to 10 monosaccharides. Polysaccharide, these were about the oligosaccharides. The last one is polysaccharides. Those carbohydrates which are having more than 10 monosaccharides, like your starch, cellulose, glycogen, gums, these are polysaccharides, and usually they are not sweet in taste, so we call them non-sugars. Now polymers, they are formed from the monomers, and when polymers they are formed, बच्चे usually यहाँ पे गलती करते हैं, they write through the hydrolysis. This is wrong, बच्चे. Polymers are formed from monomers by dehydration synthesis process. Dehydration synthesis means when water is removed. So here we are joining these two glucose molecules, and two glucose molecules, they are joined with the help of glycosidic bond, and what is removed? Water is removed. This synthesis is known as dehydration synthesis. This is clear? So there is dehydration synthesis. Now hydrolysis means, suppose I am having this maltose, and maltose is formed by glucose plus glucose. I have to break down this maltose. I will add water, and once I will add water, here now OH will come and here one H will be added, and my two monosaccharides units will be separated. So two important things: dehydration synthesis for joining, right? For joining of two monomers, for joining of two monomers, and hydrolysis for breakdown of a polymer, like you are breaking down here maltose. This process is known as hydrolysis. So structure which you have learned today, that is the glucose, this is glucose, and ribose. Now many students, when they form ribose, they put here CH2OH. Bacha, here is that if you add CH2OH here, that is our fructose, right? Here it is HOH, HOH, HOH and CH2OH. That is our ribose, C5H10O5, and fructose is C6H12O6 only. So this is the structure of fructose, glucose, and this is our ribose. And the last is about your one more example about the cellulose. Cellulose is having your 100 to 1000 glucose unit. There is one usually in your objectives for NEET, you will observe that they have given you linkages, alpha 1 for linkage, beta 1 for linkage, right? What is that linkage is about? If the orientation of this functional groups, like HOH is here, here it is HOH, these two functional groups, their orientation is different, then there will be beta. But if this orientation will be like this, here will be OH group, then when the water will be released, then we will write alpha 1 for linkage. Basically, if the both functional group, they are on the same side, they are on the same orientation, then we will use alpha. Otherwise we are going to use beta if they are having orientation like this. So if functional groups, they are not in the same orientation, then the linkage will be beta glycosidic linkage. Same, there is another polysaccharide, that is glycogen, which is made up of 1,20,000 glucose units. 1,20,000 glucose unit, right? So this is all about our today's. I'm giving you a few questions for you can practice. First question is, look at these two figures and give the correct answers. And you can count the numbers: 1, 2, 3, 4, 5. C5H10O5. It means this is our ribose. Don't make it fructose. So answer will be glucose and ribose. 21st amino acid known to us, that is selenocysteine. And identify the bonds in A and B. That you can answer me. If you have understood that what is a glycosidic bond, then answer me in the comment section, right? This is all about our today's lecture. Next we will meet you with the another revision time.